Thanks so much to Leo and the organizers for the invitation. It's nice to be here at NYU and get to share some of a little bit of what we work on in the lab, and then talk to you a little bit about one of the newer projects that has really kind of surprised me over the last few years as I move through my early career. So just to put things in context a little bit, so of course, this is probably familiar to most people in the room, but endocrine disruptors are then, of course, any chemical or mixture of chemicals in the environment that can interfere with any aspect of hormone action. Mostly in our group, we focus on these direct interactions up here. These are chemicals that are able to kind of fool our endocrine system into thinking that they are endogenous hormones, acting as receptor ligands or agonists, or can bind to and block endogenous hormone action, acting as receptor inhibitors or antagonists. But there's, of course, a whole suite of indirect interactions as well. These chemicals can interfere with receptor expression, how signals are transduced, a whole host of epigenetic alterations, which we've heard a fair bit about, all sorts of effects on hormone synthesis, transport, distribution, breakdown, and then a variety of cell fate decisions, some of which we look at in our lab. And of course, these chemicals come from a variety of places, most notably, perhaps for our talks today, plastics. So a lot of the work that we do in our lab focuses on obesity. This is, I'm sure, no surprise to anyone here, but currently about 42% of the US adult population is obese, which is a statistic that never ceases to amaze me. This is about 1 in 10 infants and toddlers, 1 in 5 2 to 19-year-olds. This schematic on the right, showing worsening obesity rates, is, we're seeing prevalence of, up to 40 or 45% in some states. This is expected to worsen by 2030. This, of course, contributes to a huge share of the US healthcare burden, a variety of comorbidities, type 2 diabetes, cardiovascular disease, hypertension, et cetera. And interventions have really produced only modest effects. And of course, this is a global problem, not just a US problem. So this idea that chemicals might be a causal factor in this was first suggested in 2002, this paper by Bailey Hamilton. And it did two things, right? So it, one, discussed how caloric intake, activity, and genetics were insufficient factors to explain the magnitude and speed of the observed trend. And then it also summarized the wealth of available animal and also some human data on 70 plus years of research on obesogenic effects of things like antibiotics, PCBs, plastics, pharmaceuticals, et cetera. And so how exactly do these chemicals play a role in this? So there are lots of different mechanisms. This is a great paper from Jerry Heindel and collaborators. Some of the things that we focus on in our lab are these processes of early precursor stem cell decisions leading to increase in adipocyte proliferation or lipid uptake. And then we're starting to look at some of these earlier decision phases as well, from these multipotent progenitor cells to the adipocyte lineage. So I'm going to talk a little bit today about one of the projects that has come out of my early career development. Something that I certainly, 4 or 5 years ago when I was interviewing for faculty jobs, did not see myself working on, but here I am. So turf, probably not something that anyone is unfamiliar with, but it's this artificial product designed to mimic natural grass surfaces. A few statistics, so there's between 12 and 13,000 artificial turf fields currently installed in the US, with more than 1,500 new installations each year. Also seeing increasing use in residential applications, right? Particularly in these drought-stressed regions. You can have this nice green backyard. Children are estimated to spend up to 12 hours a week playing on artificial turf fields. This was a point that was mentioned earlier. There are few options for recycling, right? So old turf material at the end of its useful life of between 8 and 15 years is often dumped or landfilled. And then, of course, artificial turf consists of synthetic fibers, an infill, which is a fine particulate material that's used as filler between the blades of the synthetic grass, and then a base material or padding. And so I got into this through our NIEHSP 30 Center at Wayne State. So I was introduced to these folks at the Ecology Center, which is an environmental nonprofit out of Ann Arbor. And we were talking about ways to collaborate and settled on artificial turf as something that they were really excited about and also something that had always interested me as well. And so they sent us a box loads of turf material, so both the turf samples themselves, the filler materials, right? And so, 15 years ago or so, when artificial turf was getting a lot of press attention, it was primarily focused on crumb rubber. We still use a lot of crumb rubber, but we also have a variety of polymer-coated, what we would call environmentally friendly alternatives, and even some natural options, things like walnut hulls and sand, as well as some padding samples. And so we extracted these with solvents in the lab, dried them down, and then used those kind of turf material extracts in reporter gene assays to look at activation or inhibition of specific human hormone receptors, and then starting to characterize some of the potential toxicity from those mixtures of chemicals associated with the turf. And so we settled on a bit of a subset so that we could look a little bit more broadly. But specifically, we looked at activation and inhibition of the estrogen receptor, androgen receptor, glucocorticoid receptor, thyroid receptor, aryl hydrocarbon receptor, certainly very important for the crumb rubber, and then PPAR gamma, which is often considered the master regulator of fat cell development. So just to introduce you just briefly to our adipogenesis assays, these are a variety of models are used for this. So human and mouse preadipocytes, historically most widely used, but increasingly with the commercial availability, people are shifting to human mesenchymal stem cells. And so generally we are seeding cells in multi-well plates, letting them grow, starting this process of differentiation with the adipogenic cocktail induction. We do that for about 3 days, start our test chemical treatments, and then have another 18 days where we allow these cells to accumulate lipids. And then primary endpoints of triglyceride accumulation, which is our marker for extent of fat cell differentiation, and then pre-adipocyte proliferation as measured by DNA for adipose tissue expansion. And these are some differentiated cells. You can see our kind of blue DNA dye. So these are the nuclei. And then you can see often many kind of this signet ring appearance of the lipids surrounding the cell nucleus. All right, so just a little bit about some of the turf data that we've been seeing. So this paper just got accepted a week ago or so. This is my PhD student Kyle Siegel's work. So I'm just going to show you a few little snippets from this paper, but it was just published in Environmental Toxicology and Pharmacology, so feel free to take a look. So we broke our turf material extracts into two groups, so those coming from commercial or new products, so non-weathered, and those that were coming from actual fields out in the wild, right? And those we're going to term weathered. And so these are non-weathered turf samples. So you can see this is our positive control, rosiglitazone, which induces this nice, robust positive control dose response. And then these are some of our non-weathered turf samples. So you can see very modest activation of PPAR gamma. In contrast, our weathered turf samples, you can see there's a much greater number of samples and magnitude of effect for the weathered turf samples relative to the non-weathered. So we were wondering, will that translate into kind of a functional effect on adipocyte development? Turns out not really. So we actually see a greater effect on lipid accumulation in our non-weathered turf extracts relative to our weathered extracts. Why that is, probably there's a variety of signaling pathways that induce adipogenesis, a variety of both endocrine signaling induced and others, many of which these chemicals have, and many of which are greater in the weathered samples than the non-weathered samples. And so this is not so much a PPAR gamma story as I think other pathway story. And then we didn't see any difference in the extent of proliferation between those. The other little snippet I'll show you is that turf material extracts were able to pretty robustly simulate AHR activity. So relatively modest in the non-weathered turf, but you can see the weathered turf in many cases have efficacy that exceeds our positive control, which is beta-nethylflavone. Kyle is also very interested in cardiovascular health, so he did some toxicity assessments in H9C2, so these are rat cardiomyoblast cells, and so we can see our positive control. We can see 3 high AHR efficacy extracts, so we can see an induction of toxicity, and if we then try to counteract that, right? So if we presume that AHR activation is inducing the cardiotoxicity, can we co-treat with a AHR antagonist and block that cardiotoxicity? And it turns out we can. All right, so I'm not going to talk any more about data. I'm just going to share a little bit of where we're going. So we want to see if we see conserved effects in vertebrates. So our model organism is the zebrafish. So molecular mechanisms underlying adipose depot development are highly conserved across vertebrates. We can actually map out where adipose is accumulating in the zebrafish. Unlike a lot of mammals, imaging of internal adipose in zebrafish is high resolution, very quick, easy to do. Fish adipose contains a heterogeneous cell population, including adipocyte progenitors, just like in humans. So this is a nice model for this assessment. This is what that looks like for us. So we are collecting embryos at about 1 day post-fertilization. We're exposing to chemicals, or in this case, our turf material extracts for 5 days. At 6 days, we look at energy expenditure. We look at locomotor behavior, which is a standard neurodevelopment measure. We age our fish out for another 24 days without chemical exposure. And at 30 days, we map our adipose. We look at extent of adipose development and where it's developing in visceral versus subcutaneous depots. And then we also have some early stage cardiotoxicity assessments. The other little thing that I'll just throw out as a teaser is that with the help of pilot funding from NYU here, we also created a small human cohort in Detroit. So we've recruited about 50 youth soccer players playing on artificial turf fields and natural grass fields. We've done hand wipes before and after use, and they're now with a chemist to measure diverse SVOCs and see what exposures look like between these fields. And so just in summary, I want to really all of the work that we do in our lab is around environmental mixtures. These are very complex. We still don't really well understand constituent chemical interactions. And I think it's really critically important to assess realistic whole products for mixture testing. Artificial turf, another example of a multi-component plastic product. It's layers on layers of plastic with widespread human exposures and poor understanding of hazards. Plastic turf-associated chemicals and mixtures disrupt endocrine signaling and can contribute to adipogenic and cardiotoxic effects in vitro. And so with that, I'll just thank my group. So in particular, Kyle is the PhD student that's been leading this, but both he and my postdoc, Roxanne, have been working through the zebrafish developmental exposures, and really the whole group's been helping with our human health assessment. And I'll stop there. Thank you.